Hey there, welcome to another deep dive. Ever imagine being like a genetic mechanic, taking things apart, figuring out how they work?
That's kind of fun, actually.
Right. Well, today we're diving into the world of heterologous expression. And it's basically that, taking a gene from one organism and seeing if it works in another, like trying a car key in a different make and model. Sometimes it fits, sometimes it doesn't.
And sometimes you get a completely unexpected result.
Exactly. Our subject is this 1995 paper, Heterologous Expression of Genes in Bacterial, Fungal, Animal, and Plant Cells. Sounds pretty dense, right? Don't worry, we'll break it down.
We're going to unpack why this technique, even back then, was so groundbreaking for researchers. Think of it like this. It turns scientists into genetic detectives.
Ooh, I like where this is going. Tell me more about these detective scientists.
Well, they use this thing called expression cloning to figure out what a gene does. See, you can't always tell just from the DNA sequence what a gene will do.
It's like having a blueprint, but no instruction manual.
Exactly. So they take a gene, stick it in a different organism, almost like a guest chef taking over a new kitchen.
Okay, I'm loving this analogy. You can learn a lot about a chef by how they handle new ingredients, right? So they're throwing this gene into a new environment and seeing what happens.
You got it. And the first stop on this journey takes us to the lab workhorse bacteria, specifically E. coli.
Ah, E. coli. Simple, straightforward. A classic for a reason, right?
Exactly. Scientists knew them inside and out, easy to grow, perfect starting point for some genetic detective work.
So do they have any early wins with bacteria? Any big aha moments?
Yeah. Oh, absolutely. One of the first came in 1976. Researchers got a yeast gene to work perfectly inside E. coli, even got the bacteria to produce histidine. That's an amino acid vital for life.
Wow. It's like teaching your dog to make you coffee in the morning, totally unexpected, but hey, if it
Yeah.
works. But I'm guessing bacteria, as simple as they are, limitations.
You're right. When scientists started digging deeper into plant and animal cells, much more complex bacteria just couldn't quite mimic those systems.
It's simple.
Think of bacteria as the training wheels, great for the basics, but eventually you need a more sophisticated ride.
Okay, so what did they upgrade to? What was the next step up in this genetic joyride?
This is where things get really interesting. Scientists turned to an unlikely hero, yeast.
Yeast, like the stuff that makes bread rise? Wait, really?
Yeah. Yep, the one and only. Turns out this humble organism was a game changer, especially for plant science.
I gotta know what makes yeast so special for studying plant genes. Besides making delicious bread, of course.
Well, yeast cells, while still pretty easy to grow like bacteria, share some surprising similarities with plant cells, especially how they get their energy.
Really? I would have never guessed.
It's all about something called proton gradients. Basically, they use a similar kind of battery to power their cellular processes. A bit technical, but trust me, it's a big deal.
So it's like yeast had this hidden plant DNA tucked away all along. No wonder they get along so well in bread dough, right? But how did scientists actually use yeast to study plant genes? What did that look like?
They developed this clever technique called complementation. Picture this, you have a yeast cell, but it's missing a crucial gene, like one that helps it make a vital nutrient. It's like a car with a flat tire going nowhere fast.
Okay, I'm imagining this poor little yeast cell totally stranded.
Exactly. Now imagine taking a gene from a plant and inserting it into that yeast cell.
Okay, I'm following. So they're giving the yeast cell a replacement part, but from a plant.
Right, and if that plant gene can complement, meaning it replaces the function of that missing yeast gene, the yeast cell suddenly springs back to life.
It's like swapping that flat tire with a spare and boom, a perfect fit. You found your match. So if that yeast cell starts humming along, producing that nutrient again, they know they found the gene responsible for it.
That's the beauty of it. And they weren't just limited to one type of gene either. They used this to study a whole bunch of plant genes, from enzymes to, this is important, membrane transporters.
Hold on, transporters, those are like the delivery trucks of the cell,
Yeah.
moving things in and out.
You got it.
Yeah.
And they're essential, nutrient uptake, signaling, you name it. But studying them traditionally, a real pain.
Like trying to track down a rogue delivery truck with no tracking number.
Exactly. But with yeast complementation, suddenly they had a way to pin down these elusive transporter genes. And they had some big wins. They found genes for potassium channels, amino acid transporters, and even, get this, the long sought-after sucrose transporter.
Sucrose. Okay, now my sweet tooth is perking up. What's so special about this sucrose transporter? Why were scientists so obsessed with finding it?
Well, you know how plants use photosynthesis to make energy from sunlight.
Of course. High school biology flashbacks.
Well, sucrose is the primary sugar that plants use to move that energy from the leaves to other parts of the plant. It's basically their lifeblood.
Wow, so understanding how plants transport sucrose.
Hmm.
That's huge, not just for plant science, but for things like, I don't know,
Mm.
agriculture, food production.
You're catching on. This was a major victory, and it all came down to this elegant technique of using yeast to crack the plant world's secrets. But of course,
Wait a minute, I know that tone. There's a but coming, isn't there? There's always a but in science. What's the catch with our little buddy yeast? We're back. Last time we were hanging out with our little yeast buddies, seeing how scientists use them to crack the code of plant genes.
yeast. They're like the superheroes of the lab. Small but mighty. But even superheroes have their limits.
Right, like that one superhero who can't see through lead walls or something.
Exactly. And for scientists digging deeper into the mysteries of genes, yeast could only take them so far, especially when it came to those really intricate processes like cell signaling or how diseases develop. They needed something more. Well, more like plants and animals.
So they needed to upgrade to a more lifelike model system.
Precisely. Enter the animal kingdom.
Whoa, okay. That took a turn.
Yeah.
We're talking lions and tigers and bears. No, wait, frog eggs. That's what you said last time, right?
Frog eggs, you got it. Specifically Xenopus oocytes. They're a classic for a reason.
Okay, now I'm picturing a scientist with like a tiny little lab coat peering into a frog egg under a microscope. What's so special about Xenopus oocytes?
Well, for starters, they're relatively large, easy to manipulate. They're cellular machinery, great at expressing genes from other organisms.
So you can basically insert a gene from something else and the frog egg will just read it, like follow the instructions.
Exactly. But here's the really cool part. Xenopus oocytes are like miniature electrophysiology labs.
Electro what now?
Electrophysiology. It's how scientists study the electrical activity of cells. Remember ion channels, those tiny gateways that control the flow of ions in and out of cells.
Right, right. Those are important for pretty much everything a cell does, if I remember correctly.
Exactly. And with Xenopus oocytes, researchers could actually study these ion channels in action, see how they respond to different stimuli.
So these frog eggs were like tiny voltmeters letting scientists eavesdrop on the electrical conversations happening within a cell. That's pretty wild.
A perfect way to put it. And the paper we're talking about today, it actually highlights some fascinating research that used Xenopus oocytes to study potassium channels.
Potassium channels. Those are important for plants too, right? Like helping them regulate water balance and respond to stress. I vaguely remember learning that somewhere.
You're right. And what these researchers found by studying plant potassium channels in frog eggs was that they had some unique electrical properties compared to those found in animals.
So even though both plants and animals use potassium channels, they've evolved slightly different ways of doing it.
Exactly. It was a clear sign that plants had come up with their own solutions to regulate these channels and adapt to their environment.
That's really fascinating. It's like comparing the electrical systems of a house and a spaceship. Both use electricity, but they've been wired in completely different ways.
Love the analogy. But let's hop off the Frog Egg Express for a minute. We've got other animal cells to explore.
Right, right. Insect cells. Those were on the list. How did those fit into the picture?
Insect cells are interesting. They bridge this gap between simpler systems like bacteria and yeast and the more complex world of, say, mammalian cells. They're more adept at folding and modifying proteins in a way that resembles what happens in higher organisms.
More sophisticated, but still relatively easy to work with.
Right. That's why they became so valuable for studying certain types of proteins, especially those involved in complicated interactions like protein interactions or signaling pathways. They're also great for, get this, mass-producing protein.
Really? So it's like having a protein factory right there in your lab.
Exactly. Let's say you're studying a protein, you want to figure out its structure, maybe test how it interacts with potential drugs. The more protein, the better.
Makes sense. So insect cells, they were like the workhorses of protein production. But I can't help but feel like we're saving the best for last, right? What about mammalian cells?
Ah, mammalian cells, the stars of the show. Definitely the most complex system we've talked about so far.
The divas of the cell world, with their high maintenance requirements.
Maybe a little bit, but that complexity also makes them incredibly valuable, especially when studying genes from mammals, including us.
Right. I mean, it makes sense to study human genes in, well, human cells, if you can, or at least something closer to human than a bacteria or a yeast cell.
Yeah.
But what about plant genes? Why on earth would you study those in mammalian cells? It seems so counterintuitive. Yeah, it's like if you want to learn how to bake a cake, you don't exactly go study a car engine, right? So why study plant genes in something as complex as a mammalian cell?
You're right, it does seem a bit backwards at first, but remember those water channels we were talking about? The ones that help move water across cell membranes?
Yeah, super important for plants, obviously.
Well, the paper actually talks about this really cool example where scientists used mammalian cells to study these very plant water channels.
Okay, now you've got my attention. How does that even work?
Well, they were having a tough time studying these channels using their usual methods, you see. Difficult to isolate them, difficult to really analyze them in plant cells. So they had this brilliant idea, why not use mammalian cells as basically mini factories to produce a ton of these plant proteins?
No. So they're basically
Yeah.
putting the plant's recipe into the mammalian cell's kitchen and saying, okay, make this.
That's a great way to put it. And the remarkable thing is, it worked. Because at the core, the way that cells read DNA and build proteins, it's remarkably similar across different life forms.
So even though plants and mammals seem worlds apart, there's this underlying common language in their cells.
Precisely. And the beauty of using mammalian cells is that they're remarkably good at what they do. They're like expert protein folders, making sure everything is assembled correctly, even if the instructions came from a plant.
So it's like sending a piece of IKEA furniture to a master carpenter. They might be used to building fancy mahogany desks, but they can still put together that flat-pack bookshelf without a problem.
Exactly. And the result, they had tons of these functional, perfectly formed plant water channels ready for analysis, something they couldn't easily get before.
That's incredible. It really goes to show nature is full of these amazing connections, isn't it? Even between things that seem totally different on the surface.
Absolutely. And this research, even though it was done back in 1995, it was a sign of things to come. Scientists were starting to realize the full potential of heterologous expression. And then along came CRISPR.
CRISPR. It's like the rock star of gene editing these days. Everyone's talking about it. How did CRISPR change the game for heterologous expression?
Think of it like this. Before CRISPR, inserting a gene into a host cell, it was a bit of a shot in the dark. You hoped it landed in the right spot, that it worked the way you wanted.
A bit like sending a letter and just hoping it reaches the right person. No address, no guarantee.
Exactly. But CRISPR gave scientists an address, so to speak. They could target exactly where they wanted a gene to go, ensuring it integrated correctly and do its job.
Wow, so CRISPR took heterologous expression from a sort of hope for the best approach to a precise, controlled technique.
Precisely, and that has revolutionized the field. We're now able to study genes and their functions in ways we never thought possible.
It's amazing, isn't it? We've gone from those early experiments with bacteria to the elegance of yeast, the complexity of animal cells, and now with CRISPR. The possibilities seem endless. Who knows what incredible breakthroughs are just around the corner? Well, that's all the time we have for today's deep dive. Thanks for joining us as we explored the fascinating world of heterologous expression.